Hello, Mr. Allman here, and this is your third installment of our tour of the cell. In this short video, I want to highlight the role of the cytoskeleton and other forms of structure and support for the cell. Now, the cytoskeleton is a complex structure that extends throughout the cytoplasm. There are three main components of the cytoskeleton. Each of these are three different types of proteins that are able to polymerize and form these really long fibers that act very much like the steel support structure within a skyscraper. They all have different functions, they have slightly different structures. The names of these things aren't necessary for you to learn, however, you should be able to understand the range of functions of the cytoskeleton. So, if we look at the cytoskeleton as a whole, its most important function is structure. This is what holds the shape of the cell together, particularly for animal cells, which lack a rigid cell wall. Without the cytoskeleton, there wouldn't be any way for the fairly fluid cell to be able to maintain a particular solid shape. So, additionally, the cytoskeleton is going to anchor all the different organelles with each other. So the mitochondria, the Golgi, the ER, the nucleus, all of those things are going to be interconnected by a highway of the cytoskeleton, allowing these things to be able to maintain their position because each of those organelles are going to be in a spot in the cell that's going to maximize their position relative to their neighbors, because in many cases materials are moving from one organelle to another. So, for example, the nucleus needs to be anchored to the endoplasmic reticulum because RNA that is copied in the nucleus needs to travel out to the ER where proteins are going to be made. So, maintaining that position is going to allow the cell to maintain peak efficiency. It's also important in terms of motility. So, some cells have the ability to move. When we look at single-celled organisms, things like the amoeba are able to use the cytoskeleton to physically move from one place to another. And while most of our cells don't show motility, white blood cells are one example of a type of cell that is able to change its shape so that it can move to places where an infection has occurred. Within the cell, within all cells, organelle motility is something that does happen, particularly if you think about the movement of vesicles from the ER to the Golgi and to the plasma membrane. Finally, an important function is the movement of chromosomes during cell division, and this is something that we'll cover more in depth as we look at meiosis and at mitosis. Here you can see a more specific example of an object called a motor protein and how it is going to allow vesicles to move along the microtubule. The microtubule is the strand that you see along the bottom of this particular electron micrograph. It is the largest of the different types of proteins that make up the cytoskeleton. Along it is a vesicle. A vesicle, again, is a small membrane-bound sac that's going to have some type of material that needs to be transported within the cell. In this case, ATP is used as a source of energy so that a motor protein will move along the length of that microtubule so that that vesicle can get carried from one place to another. So these little bags of chemicals don't float around and just randomly move from one place to another, but rather you can consider, in this case, the microtubule of the cytoskeleton as the highway along which these materials are going to be transported, and those highways are going to connect one organelle to the next. The next part are the actin filaments of the intermediate filaments. And these are important in terms of movement. Those in HL biology are going to learn about the motion of actin and myosin as part of muscle contraction. Again, these are elements of the cytoskeleton. You can see the amoeba and how the amoeba is able to move by extending its cytoskeleton out in the direction that it wants to go, and then breaking down the cytoskeleton behind it, and that's going to cause a flow of cytoplasm in the direction that the amoeba needs to go. The The cilia and the flagella are two structures that are going to be extensions of cytoskeletal proteins beyond the traditional boundary of the cell. These are more specialized structures that are mostly useful for motility. They're made up of the same groups of proteins, and as you can see in the example here with sperm cells, they're going to be what allows the cell to be able to move. It's the back and forth motion of the proteins within that flagella structure is going to be what allows the sperm to be able to swim through the reproductive tract towards the egg. So the function here is motion through space. Most cells in humans don't have either cilia or flagella. The cells that do are more the exceptions, the sperm being the only one with a flagella, and the cells that line your respiratory system are one example of those that have cilia. These structures are very common on prokaryotic cells and other single-celled organisms that use them as a way to be able to allow them to move, particularly through water. The centrioles are a pair of structures that are going to organize the cytoskeleton. These structures only appear in animal cells. The centrioles are going to be a bundle of cytoskeletal elements that have this circular or star-shaped pattern, and they appear at 90-degree angles to each other within the cell. Along the boundary of the cell, we find the membrane. The plasma membrane is a phospholipid bilayer. It's two layers of fatty acids connected to phosphate heads. The details of this structure are very important for understanding transport of materials into and out of the cell, and are going to be the subject of a separate video. The function of the plasma membrane is to act as that boundary separating the cell from its surrounding environment. It's going to be selective in allowing certain materials to get into and out of the cell, and it's going to have structures that are going to allow for communication between the cell and its neighbors or the environment. Finally, I'll mention briefly some structures that aren't commonly found in animal cells. However, they are going to be significant towards your understanding of some lab work that you're going to do later in this unit, particularly the cell wall. The cell wall is going to be a cross-linked network of structural polysaccharides. Polysaccharides are going to be polymers of sugar molecules. These are structural because the way that they're linked together is going to form a very tight and a very strong network that is going to provide some important functions for the cell. As we look at the different types of polysaccharide networks, we have first cellulose. Cellulose is going to be, again, a polymer of glucose, which is a very simple sugar. And these are going to be found in all plant-like cells, whether they are algae, other photosynthetic protists, or the cells within plants. This acts as the outermost boundary. The plasma membrane is inside of the cell wall. The cell wall is the outermost part that would be visible on that cell. It is an important structure that allows plants to be able to grow upright, and so that provides them the strength that keeps them from falling over. Fungus also have a cell wall, but they have a cell wall made of a structure called chitin. And so chitin is a different type of polysaccharide. Again, it is still made up of a polymer of sugar, but it is going to be distinct from the type of molecule that's found in cellulose. One important difference is that chitin is something that we are capable of digesting, whereas the cellulose in plant cells we are not capable of doing so. And finally, peptidoglycan. The structure of peptidoglycan is shown there. This is another polymer of sugar with a slightly different structure, and it makes up the outer wall of some types of bacteria. For example, the E. coli bacteria, of which you can see here. That peptidoglycan cell wall is important because it is something that we use to distinguish between different types of bacteria using a particular type of stain that attaches to this particular molecule. So these all have the same function. They provide structural support for the cells. Cell walls are non-living. They are not considered a living part of the cell because we would say that the plasma membrane, which is inside of it, is really where the cell begins. Thank you for watching, and please let me know if you have any questions.